Hi, my name is Sara McSweeney, and I studied increasing the desiccation tolerance of Arabidopsis teff through exogenous application of abscisic acid to ensure food security for the future. A 2018 United Nations study found that we are losing 23 hectares of arable land every minute due to drought and desertification. This is a projection of what the world is going to look like in 2050, where all the areas in red is all of the arable land that we will lose by then. As you can see, most if not all of the agricultural land currently being used will be unusable by then. So what we need are plants that are able to withstand their yield while needing less water. An example of this is Arabidopsis teff. It is a staple crop for Ethiopia and South Africa and extremely valuable to agriculture because it is gluten-free and very nutritious. Arabidopsis teff is drought tolerant but not yet desiccation tolerant. Natuli in 2012 defined drought tolerance as being able to survive drought with a high internal water content, while desiccation tolerant as being able to survive drought with a low internal water content. Desiccation tolerance is preferred because the organism will be able to survive drought for a longer period of time. An example of an organism that is both drought and desiccation tolerant are resurrection plants, and this is potentially due to abscisic acid, which is a plant hormone that is known to confer tolerance to abiotic stresses, in particular desiccation. Costa et al. In 2017 compared orthodox seeds, which are seeds that can withstand extreme temperatures, to resurrection plants. And what they found was that by pre-soaking a group of orthodox seeds in an ABA solution, these seeds then maintained their ability to become desiccation-tolerant plants in the future, while the group that was not pre-soaked lost that ability after germination. They also found that the mechanisms of desiccation tolerance in orthodox seeds closely resembled the mechanisms in resurrection plants, indicating that potentially resurrection plants have the best mechanism to survive desiccation. Gaff and Levies in 1984 exogenously applied abscisic acid to boa constrictor leaf, and what they found was that this raised the endogenous levels of ABA inside the plant. This means that it is possible to change a plant's internal ABA level, and this could potentially contribute to increasing its desiccation tolerance. The gap in the knowledge is that there has been no research done on increasing desiccation tolerance of teff, and the purpose of the study was to find evidence for a possibility of increased desiccation tolerance in teff. It was hypothesized that exogenous application of ABA to teff would indicate traits that correlate with an increased desiccation tolerance. A brief overview of my methodology. It was conducted in two phases. Phase 1 was a proof of concept and optimization done in the US using the model organism Arabidopsis thaliana. Phase 2 was the experimental procedure done in South Africa using the experimental organism Arabidopsis teff. Now, Arabidopsis thaliana was the model organism used due to its small and completely sequenced genome. Any knowledge that we gain from Arabidopsis can be easily transferred to a variety of other crops. Arabidopsis teff is considered an orphan crop, meaning that it is not internationally traded and therefore gets less research funding, and it is also considered a precious resource. So as to not waste any precious resources, I first conducted a proof of concept study, and then I also didn't get access to these seeds until I went to South Africa. Phase 1 was soaking the Arabidopsis seeds in different ABA concentrations and then measuring the germination rates and viability of the seedlings after 14 days. And what I found was that there was significantly lower germination when comparing the expected rates to the observed rates. And of the few that actually were able to germinate, even fewer were alive after 14 days. So it was determined that seeds are not the most effective vector for ABA delivery, potentially because of the barrier that the seed coat created. So my mentor and I decided to use cali instead, and a callus is just a mass of undifferentiated cells. This way, there is no barrier, and this would allow an easier uptake of ABA for the plant. Phase 2 was the pilot study done in South Africa. I first cultivated the Arabidopsis teff seeds into a callus-inducing medium so that a callus would be created. I then applied the different treatments, so either a distilled water treatment, a 20 micromolar ABA solution treatment, or no treatment at all. Then the calli were cultured into a regeneration medium, which would allow the calli to develop into seedlings. These seedlings were then grown for 7 days. After that, they were dried under a laminate airflow for 48 hours. And then I performed 3 different stressor assays to them: relative water content, electrolyte leakage, and chlorophyll and carotenoid content. I then performed a ANOVA test to test for significance. Relative water content, there was a significantly higher relative water content in the ABA treatment compared to both the water treatment and control, indicating that ABA potentially helped the plants retain more water inside their cells. In terms of electrolyte leakage, there was significantly lower in the ABA treatment compared to the water treatment. And electrolyte leakage is an indication of the structure of the cell membrane. So a lower electrolyte leakage means that the cell membrane is more structurally sound. So potentially ABA had a protective effect on the cell membrane. In terms of total chlorophyll and carotenoid estimation, although there was no significance, there was an overall trend that the total pigment concentration was highest in the ABA treatment, so potentially ABA had a protective effect on the photosystems, which allowed for a higher pigment concentration. So the hypothesis was supported. Aerugos's TEF cali that were exogenously applied with ABA did exhibit traits that are associated with increased desiccation tolerance. The results from my study were consistent with Costa's, and that showed that soaking does mean that traits are correlated with an increased desiccation tolerance, although I found seeds not to be the most effective vector of ABA delivery. This was a trait study that did show increased desiccation tolerance, but the seedlings were not fully grown, so this could be a potential limitation. Also, the cali were not the most ideal vector because they do need to be induced and it is difficult to mass produce. So potential future research would be to find a tissue that is more suitable for mass production. Overall, though, ABA does warrant further study because it is a way to increase the desiccation tolerance. For future research, I would like to identify the ABA pathway in TEF and the genes involved with desiccation tolerance of TEF so that we could potentially use this in other non-desiccation tolerant crops. If we're able to increase the desiccation tolerance of TEF even more, it could have an even larger role in agriculture than it does today. It could be a food source for even more countries, and we could then potentially use otherwise unsuitable land that traditional crops could not survive in. These are my references. Thank you very much.